Good morning and welcome to today's MPG primer session. We are very excited to have three speakers today. The speaker who will be coming from the MPG portal side is Dr. Maria Constanzo. She's creative lead of MPG portals. Portals are available for multiple complex disease areas and are aimed at catalyzing the clinical translation of genetic discovery. Maria's role on the portal team includes collaboration with data generators to select and aggregate the most authoritative data sets for the portals, participation in tool and interface development, and outreach to spread awareness and understanding of the portals. Prior to joining the Broad in 2015, Maria was a biocuration scientist with the Saccharomyces candida aspergillus genome databases at Stanford University and performed basic research on the genetics and molecular biology of yeast at Cornell University. Preceding her discussion of portals will be two content experts, Dr. Tobias Walther, who's a Howard Hughes Medical Institute investigator, professor of molecular metabolism at the T.H. Chan School of Public Health, and professor of cell biology at Harvard Medical School. He is joined by Dr. Robert Farese Jr., a professor in the Department of Molecular Metabolism at the Harvard Chan School of Public Health, professor of cell biology at Harvard Medical School, and associate member of the Broad Institute of Harvard and MIT. Drs. Walther and Farese have run a laboratory jointly at Harvard since 2014. Through their work together, they elucidate the molecular mechanisms for triglyceride synthesis and storage in lipid droplets, or LDs. They uncover fundamental mechanisms of LD formation, LD expansion, protein targeting to LDs, and cellular lipotoxicity when triglyceride storage in LDs is overwhelmed. They elucidate the physiology and pathologies linked to oil synthesis and storage in LDs, including discovering mutations in these pathways that lead to congenital diarrheal syndromes or lipodystrophy in humans. So we are really excited for this cross-disciplinary talk today, starting first with the scientific content and followed by a tour of portals.
Well, thank you so much for the introduction. It's a pleasure to be here. And I'm looking forward to introduce to you some of the science behind the portal. And then Maria, I believe, will give you an introduction of how to best use this. Before I get started, I want to first acknowledge that sort of all the work that goes into the portal from the science side is really stemming from a scientific partnership between Bob, who's also on the call, Bob Farese, and myself. I've been running a joint laboratory now for well over a decade. And the portal in specifically, of course, integrates data from many research groups, but particularly also is catalyzed by the work of Noel Burr, Jason Flanagan, and Maria Constanzo, and also DK Jung particularly played an important role in sort of its design. And we're very grateful for that and generally the collaboration with the Broad Institute to get this going. Now, in the first part, I will give you a brief introduction in sort of the basic biology that motivated the development of this portal. And just to start out, I want to remind you that a absolutely fundamental, crucially feature of life is that it is driven by metabolic energy flows through biological systems. And only when energy flow through systems are we able to beat entropy, generate structures, etc., and do all the wonderful work that we're interested in doing. And of course, metabolic energy for this flux is not always available readily. And often there are fluctuations in this availability. And as a consequence, pretty much all biological systems have evolved the capacity to at least temporarily buffer for these fluctuations and store metabolic energy. And by and large, this occurs in the form of lipids. You can see on this slide here, roughly drawn to scale for a healthy human, that most of the metabolic energy is stored in lipids and only a smaller fraction is stored as carbohydrates. And of course, this is crucial to allow for expanding energy when needed, for instance, when you want to perform work, want to stay warm, etc. Now, these lipid stores predominantly occur in the form of a molecule called triacylglycerol. It's called triacylglycerol because it has a glycerol backbone onto which 3 acyl chains are esterified. Now, the chemical structure of this molecule makes it very, very hydrophobic. It makes it a neutral lipid. It doesn't carry a charge. And of course, you all know what happens when you mix an oil such as this with water. The two phases do not mix. Instead, phase separation occurs and oil droplets form. You can recapitulate and reconstitute this process, for instance, in a cocktail glass. I might have in the backyard of on the porch of my backyard, and you just put oil and water together. And you can see that the two don't mix. It's pretty straightforward, but it really generates a fundamental problem for cells because now cells store the metabolic energy in an oil phase that is segregated from water, where life really happens in all the biochemistry that we know. And at its interface, actually, there is a highly denaturing surface that has the tendency to really take apart proteins. So cells have evolved complicated mechanisms to deal with this problem. And these mechanisms are the research focus of our laboratory. And as you can see in cells, the little droplets are actually not making one big clump, but instead form organized droplets in cells that with their presence, cells are converted to emulsions, where the cytosol forms a contiguous phase and the lipid droplets a dispersed phase. In order to prevent actually this sort of segregation in just two distinct phases, an emulsifier, surfactant, usually phospholipids, keep the two phases apart. This does the system with peculiar physical properties, and one of them is that lipid droplets in a cell diffract light. And as a consequence, lipid droplets were among the first organelles discovered, here shown in this beautiful drawing from Richard Alt, a German pathologist, who found in frog liver hepatocytes that lipid droplets are an extremely abundant organelle. Incidentally, this is the same person who also coined the term nucleic acid and discovered mitochondria in similar drawings. Lipid droplets are present, as far as we know, in every kingdom, and they can be found in bacteria, in flies, and yeast, and mammals, and they're so stained here either in this yellow color or in this green color. And as far as we know, also pretty much every cell type that we've looked at has the capacity to store lipids. On a molecular level, lipid droplets are highly unusual. Unlike other organelles that are bounded by a bilayer membrane, lipid droplets are bounded by a monolayer of phospholipids that covers the core of neutral lipids that contains the triacylglycerols and other neutral lipids such as sterol esters and maybe retinol esters and other hydrophobic molecules. We now also know, as you can see on this micrograph here, that lipid droplets can interact tightly with mitochondria and the endoplasmic reticulum and are really very dynamic hubs functioning in metabolism. Importantly, pretty much all of the cellular physiology on lipid droplets is mediated by proteins that specifically target the lipid droplet surface. Now, in humans, we all know, of course, that a lot of the fat is stored in adipose tissue, but it's important to note that also pretty much every other tissue has the capacity to store lipid droplets, and some do so prominently. After a meal, for instance, the intestine temporarily stores lipid droplets. You can see this here in blue. When adipose tissue mobilizes fatty acids, a lot of them are temporarily stored in the liver. You can see this here in red. And then there is other participation of lipid droplets in interesting physiology. For instance, the heart runs predominantly on fatty acids, and in heart pathology, often lipid droplets accumulate. And milk production, of course, leads to a lot of secreted lipids, and this is a very poorly understood but fascinating process in which lipid droplets are somehow extruded from mammary epithelium. Now, as I mentioned, the main function of lipid storage that you think about, of course, is to put the excess fatty acids in the temporary storage form. In modern societies, often this occurs to a very large extent, leading to a current pandemic in obesity. What is a little less understood is that what happens in this case is that it is thought that when this capacity of lipid droplets to accommodate fatty acids, as occurs, for instance, in obesity, now excess fatty acids accumulate in other lipid metabolites, and that is what really triggers disease linked to obesity, because, for instance, it can lead to stress of the endoplasmic reticulum where lipids are synthesized, and either adaptation of cell death of these cells. And depending a bit on where this exactly occurs, different pathology manifests. So for instance, linked to obesity, very common is fatty liver disease, and this has become a problem of epic proportions and a huge unmet medical need. Also linked, of course, are diabetes and insulin resistance when lipids accumulate in the pancreas or the muscle. For other lipids, for instance, sterol esters, their accumulation is tightly linked to the development of atherosclerosis as I mentioned, when there is heart dysfunction and cardiomyopathy, very often lipids accumulate. I already mentioned that particularly fatty liver disease is a massive problem, not only in the United States, but really globally, to the point where 1 in 4 people now has fatty liver disease. Now, what is a bit less known is that there's, of course, other reasons why a similar pathology can arise. One of them is that if there are defects in the processes of lipid storage, for instance, the formation or catabolism of lipid droplets, so that similarly as in obesity, now the excess fatty acids can't be stored efficiently, instead accumulate as metabolites, lipid metabolites, and trigger stress pathways that ultimately lead to pathology. For instance, then, lipodystrophies are diseases in which lipid droplet formation is impaired, and because of that, excess fatty acids spill over to other tissues. And while this person here shown on the right might look relatively healthy to you, it shares many of the pathological features of obesity, because this person is deficient in a gene that is needed for normal lipid droplet formation, and as a consequence cannot form or maintain functional adipose tissue. Now, I mentioned already, lipid droplets were discovered in the 1800s, but then, to be honest, largely forgotten, because people actually considered them somewhat inert stores and bubbles of fat that probably aren't that interesting. So this has changed, however. And when you look at sort of the publication trail for lipid droplets, you can see that sort of just starting somewhat randomly after the Second World War, not much happened initially. Then there was a little bit of activity that I think particularly should be here. In 1993, the first lipid droplet protein was identified that argued that there is a specific set of proteins that is bound to lipid droplets, and as a consequence that this might be actually more than just a bubble of fat. And I think the field really took off when we started to be able to do the first genetic screens for phenotypes associated with lipid droplets and identify genes that are involved with lipid droplet biology. Now, since that time, our laboratory and other laboratories have performed a number of these screens. And I'm just going to show you one very briefly in which we used, in this case, human macrophages to identify phenotypes associated with lipid droplets. In this case, this was done as a high content screen using imaging, in which we depleted systematically every gene in the human genome and asked which of those genes have an effect on the stores, on lipid droplets and lipid droplet storage. And you can see here that these genes fall into distinct classes where each class has a somewhat unique phenotype. For instance, this first class here has very small lipid droplets that are dispersed throughout the cytoplasm, whereas you can see this fifth class here looks very different and has a few and larger lipid droplets compared to the control. Just to give you a little bit of insight that, of course, we don't stop there. One of the genes in this fifth class here is called SAPN, and is actually what's mutated in the patient I had shown you before. And just very briefly, our lab and many others have contributed to understanding how this protein really works. And it turns out that it forms a large cage in the endoplasmic reticulum. We recently solved the cryo-EM structure of this protein. And this cage structure is intimately involved in the formation of lipid droplets. And if you just look at the process, how this occurs, we now know that there are enzymes discovered initially by Bob Farese and cloned that synthesize triacylglycerol from diacylglycerol and fatty acids in the ER membrane. And that this complex, which was identified through genetic screens in yeast and other systems, has the capacity to catalyze phase transitions and promote the initial formation of a lipid droplet from the endoplasmic reticulum, as you can see in this animation here. Sorry. This is just one example of how the screens and the follow-up work on really the mechanistic underpinnings of lipid droplet formation have led to a better understanding of disease. I don't have time to go through this, but we now know that there is a whole slew of rare monogenetic diseases that are linked tightly to the process of lipid droplet formation and either result in, for instance, predominantly lipodystrophies and forms of lipodystrophy, or often also in diarrheal syndromes because our intestine is confronted with an influx of fatty acids right after a meal. And if lipid droplet formation is compromised in that tissue, often tissue dysfunction ensues. I don't have time to go through all the details of this, but importantly, I mentioned that after a lipid droplet forms, much of its metabolic function are attributed to proteins that localize to the surface of lipid droplets. The first such protein was discovered by Dean Lawness and Andy Greenberg and named perilipin for being around lipid droplets. Now, that had motivated, again, proteomic screens to identify the protein sets of lipid droplets, again, in our lab and many others, to the point where now there's more than 500 proteomic studies of lipid droplets. Oops. These have collectively identified some proteins that you would expect there and that appear to be there in every lipid droplet. This include, for instance, enzymes involved in the synthesis or remodeling of lipids or their mobilization, such as lipolysis. Importantly, though, there are many, many proteins for which we either don't understand their function at all, or we don't understand what they're doing on lipid droplets. For instance, we found transcription factors that temporarily hang out on lipid droplets, where this localization appears to be important to coordinate the physiological response to lipid storage. And again, this is just a short list. Many of these proteins that are found in lipid droplets are now known to be risk factors or monogenetic drivers of metabolic disease. And I just want to point out particularly two of them, PNPLA3, and which is a major risk factor for hepatic steatosis and a drug target worked on by many companies right now, as well as HSD17B13, which is particularly important to actually predispose people for the transition from fatty liver disease to more severe forms of liver disease, such as NASH and hepatocellular carcinomas. Now, with what I've just told you in the last couple of minutes, you might have already realized, okay, there seems to be quite a bit of data out there. Some of that has been generated systematically, and some of it has been generated sort of more traditional way. And this is really a motivation to generate the lipid droplet knowledge portal. In this portal, as Maria will demonstrate, we've tried to incorporate some of the most reliable high throughput data that's available and that was systematically generated, annotated with as much as we can about the function of this protein as it's known. And we are linking those data to human genetics, to human genetic data that allows you to cross-reference, for instance, which of lipid droplet proteins have been linked to a particular disease or go the other way around, starting, for instance, from phenotypic descriptions that have been already widely used by the portal community. I also want to stress that this is a living and breathing portal, and we're constantly adding to this lipid droplet portal. Both work from our lab and from other laboratories in the community as it becomes available. Just to give you an example, of course, one of the questions that you might ask is like, okay, how do these proteins get to lipid droplets that are sitting here? And if you want to particularly prevent their accumulation, as is a goal for some of those more pathogenic proteins, you could do a screen to ask that. And so we recently completed a genome scale screen, again, using high content screening to identify proteins that abolish or that are important for targeting of such a metabolic enzyme to the surface of lipid droplets. And with that, you can see, you can ask, okay, which genes are particularly important, because once you abolish them, the amount of protein on the lipid droplet is dramatically decreased compared to the distribution of all hits in such a screen. And you can find in this case that there is membrane trafficking machinery, maybe as you would expect. My whole point of this is not to go through the specifics of this story, but to really tell you that this hopefully will be an expanding resource that is also tightly interlinked to the other portals, such as, for instance, the cardiovascular portal or the metabolic syndrome portal. I'm already at the end here of this very brief introduction and want to just briefly mention that, of course, this is only possible because we have a fantastic group of investigators working with us. And today, I particularly want to mention, as besides the portal people, Niklas Meyer, who's now at the Karolinska Institute, who's been driving a lot of the integration of the portal, and also Lina Kuruvila, who's done an initial screen that was done here, as well as Natalie Kramer, who did a lot of the initial proteomic works, as well as some of the other people in our lab who contributed here, and for the last piece, particularly Jion Sung. And of course, this work is funded by institutions that are kind enough to give us money, and particularly want to highlight the NIH, NIGMS, NIDDK, who have funded Bob and me, as well as the Howard Hughes Medical Institute. And with that, I'm going to hand it over to Maria, unless there is any Q&A time, or Bob has anything to add that I butchered badly here. And thank you for your attention.
Okay, so thanks to Toby for that great introduction, and I'm going to show now how the portal, how the science is represented in the portal, and I hope Bob and Toby, you will jump in if I misrepresent any of the science, because this is not my expertise. But so what I'm going to do today is to just go over the portal, demonstrate what's in it and how you can use it. And then I want to pivot a little bit to talk about a new service that we have that actually grew out of this whole Lipid Droplet portal project, and that I think could be pretty useful for members of our MPG community. So let's see, I'm going to come back to the slides later. And right now I'm going to do a live demo because the portal is so lovely and fast. That's an easy thing to do. So the portal is found at lipiddroplet.org. And before I get into the scientific part, I just want to point out that there are some informational pages here. There's an about page that lists the team. And yeah, I want to acknowledge all of my colleagues on the portal team, especially DK Jang, who basically built this portal pretty much single-handedly as far as the engineering goes. And of course, all of our collaborators in the lipid droplet world. We had a lot of fun working on this project. There's also a data download page where they have made all of these data available for download in files. So that's really nice. So when you're coming to the portal, there are basically two ways that you might want to approach it. You might want to come with a particular gene of interest in mind and find out everything that's known about that with regard to lipid droplet biology. Or you might just want to, you might come with a set of criteria of genes that could be of interest, and then look for genes that answer those criteria. That would be using the gene finder tool. So I'll start by showing the gene pages and then move to the gene finder tool. So when you come in with a gene of interest, you just type it in the entry box, and this takes you to the gene page. At the top of the gene page, there's a sort of a traffic light icon here that gives you at a glance an idea of what's going on with this gene with regard to lipid droplet biology. So the green traffic light shows the results from the systematic RNA interference knockdown of all genes that they did to see whether that knockdown affected lipid droplet parameters. And so if it's green, that means that knockdown of this gene did affect lipid droplet parameters. The LD's traffic light asks whether this protein, or shows whether this protein was found in lipid droplets in a bunch of different cell types, or if the homologous protein was detected in mouse liver lipid droplets. So if this light were green, it would mean that that protein was found in lipid droplets in all 4 of these human cell types, and in mouse liver, it's found in lipid droplets in those cells. If it was yellow, as it is here, the gene product is found in one of those experiments. And if it were gray, that would mean it was not located in lipid droplets. So this section here shows the results of that high content image analysis for images of cells in which each gene was knocked down with RNA interference. So there are 5 sort of categories of parameters, and the vertical line on each shows you where this gene falls in terms of those parameters, which are measured by an RZ score. So for example, knockdown of this gene led to higher intensity of lipid droplets. And if you want to know all of the details, you can expand it to show the granular parameters that go into each of these categories. And you can look at these as either a histogram or a box plot. And if you mouse over each one, there's a little bit of explanation as to what that is. So then you can see the actual images that led to those, from which those parameters were derived. And so you've got control versus knockdown, and you can explore these, and there are tons of images. If you can really get into this, they did many replicates, and you can look at lots and lots of images. Now, one really interesting aspect of this is based on all of this image data, you can cluster genes in terms of how similar are their patterns of all these parameters. And I'm going to switch to another gene to show an example of that. PSMA1 is a subunit of the proteasome, and this is a large protein complex molecular machine that degrades proteins. And so when you look at the clustering, you can see that many other proteasome subunits cluster with this gene rather. They have the same pattern of response to RNA interference knockdown. So that tells you something interesting, that, it suggests that the proteasome could really be involved in lipid droplet formation or morphology. And in their paper, Toby and Bob look at other clusters, and you can really draw a lot of conclusions from the co-membership in a cluster. And so this clustering is shown on all the gene pages of the lipid droplet portal. I'm not going to go in detail through all of these. There's a lot of work here. All the papers are referenced in the portal, but there's transcriptional regulation by acetylated LDL. You can see whether the protein is enriched in the lipid droplet fraction. Now, here in this protein that we're looking at is not enriched in lipid droplets. So this section shows 4 of the experiments that went into that traffic light display at the top. And then there's a whole section on experiments in mouse proteomes. And you can see whether you can look at the organelle of your choice and see graphs about whether how this responded under low or high fat diets, or a change from low to high fat diet. And also if there are any phosphorylated sites, and whether those change when the diet changes. Let's see. Oh yes, I wanted to show, let me switch to another gene. Oops, RAB1A is what I want. This protein is localized in lipid droplets. And we can learn a lot from model organisms, of course. And at the bottom of this page, you can see Drosophila orthologs of a protein. And in this case, if you click on this, so there are gene pages for the Drosophila genes too. And this Drosophila ortholog is also strongly localized in lipid droplets. And so, yes, there is similar imaging parameters for the Drosophila genes, and lots of images to explore. Okay, and so finally, human genetics data are integrated into this portal. So I hope you're all aware that our group makes knowledge portals for genetic and genomic information for complex diseases. And so the data in this section are drawn from those knowledge portals. And these represent gene level genetic association scores calculated using an algorithm called MAGMA that just looks at the aggregate genetic associations across a gene. And so that you can see the scores here. So this gene is, it has a potentially significant association with these diseases. And I should say that MAGMA has its caveats. This doesn't necessarily mean that the gene is absolutely causal for these diseases. The signal could result from nearby associations in nearby genes, but it gives you a clue as to what might be going on, at least what's going on in the region of the gene. And then if you want to know more about the gene, the genetics and the genomics around the gene, you can click on one of these and go to the gene page in the type 2 diabetes or cardiovascular disease knowledge portals. Okay, I think I'm going to switch to the complementary perspective, which is what if you want to you don't know which gene you're interested in, but you want to set some parameters and find interesting genes. So in this case, we'd go to the Gene Finder tool. And just want to say first that there is a pretty detailed tutorial that leads you through all the steps in the tool and what the results look like and so forth. But let's just do it for fun. Okay, so you can choose these criteria. You can choose, as multiple criteria if you like. But let's just say we'll look for genes that determine lipid droplet morphology. Let's just put in type 2 diabetes. You start typing and then you can select the phenotype that you want. You can select 2 phenotypes if you want. And you can also set a threshold for p-value. So let's just set that. And so when you put in those criteria, then you get a screen that shows the criteria that you've selected. And also this string, you can copy this and then later you can just enter it to reproduce the search without having to re-enter all of the criteria. So if you want to change this, you can click edit, but if you're satisfied with it, you can submit the search. And then we get a list here of genes that, okay, so for which there are these lipid droplet criteria. So these are genes for which with knockdowns that affected lipid droplet parameters, and these are some of the scores. And then their MAGMA scores for those genes for association with type 2 diabetes are listed here. And so you can see that we've narrowed this down to 123 genes. So you can tailor this search however you want to find genes that are of particular interest to you. And then these links go to the gene pages in the lipid droplet portal, so you can then go and see the details. Okay, I think that is about all I had to say about the portal. Toby, Bob, did you want to add anything to that?
I just want to, now that you've shown this, add one thing very briefly, and that is to do sort of with the dynamic nature of the portal. Anybody can submit data for the portal, and there is on the about page there's a way how to do that, who to contact, and also there is the process then is that a scientific advisory board that is mostly external will decide on sort of the suitability of the data for the portal. This includes Joel Goodman at UT Southwestern Medical Center, Natalie Kramer at the Helmholtz Research Center in Munich, Michael Wrighton at the Karolinska, and James Olsman at UC Berkeley. And if the advisory board agrees that this is a good data set to include, then we have a process to do that. And I also want to just say that we are thankful for those people to help us do that. Bob, did I forget anything?
No, I have very little to add. I think you all summarized everything really, really well. And I would just say it's been a fantastic collaboration to build this portal. It really is an amazing effort by a whole team of people that perform different roles to get this together. And I think it's really valuable, and just highlighting how valuable it is. Our laboratory generated initially a lot of the data in the portal. However, the data are just overwhelming, right? So like even we have only begun to scratch the surface of this data, and this portal makes it so easy for us to just go in and probe our data sets and others' data sets as they're accumulating in there, and make connections and generate hypotheses. So it's extremely valuable. for this area of biology, and we hope for the area of cellular lipid metabolism in general. So I encourage everybody to try it out and then provide feedback, particularly if you see ways that the portal can be improved, because we want it to be useful and as Toby said, to be sort of a living site that really grows over time with more data and more utility. So thank you.
Great.
And thank you so much to all three of you for your contributions. This is a really fascinating set of questions and work that you're doing. Maria, I wanted to just note one thing that caught my attention on one of your earlier slides, which was, I believe, and please correct me if I'm wrong, but an association between short sleep duration and some of these lipid storage phenotypes. And it occurs to me, if I recall correctly, that there's an association just in public health data between sleep deprivation and obesity. And I wondered if any of the work that you're doing sort of gives us any insights on the mechanisms as to whether it proceeds, for example, through sleep duration and control of appetite, or something more complicated about lipid storage and a direct effect of lipid storage on sleep duration in some complicated way. So
that's a great question. And I don't really have the answer. I know that's a very active area of research. You can explore these types of connections in our other knowledge portals that have genetic and genomic information. And you can look for co-localization of genetic associations between obesity and sleep, short sleep duration, for example. So yeah, I think that between those portals and the Lipid Droplet portal, there's a lot to discover in there. I don't have the answers yet. Yeah, and I do want to mention that I have another short subject to talk about, but let's handle the questions about the Lipid Droplet portal first.
Great, thank you so much, yes, and thank you for mentioning that so we don't constrain your time too much. There is one question that's popped up on the Q&A, which I will read now. It says, on the portal, when I look into paralipins, classic LD proteins, the hit circles are gray and indicate no hit. Why is this?
Could you give us an example of one of those proteins?
I can answer that.
Great.
I think that has to do with the hit classification in this case is based on whether or not they had an effect on lipid droplet morphology. And it turns out that although paralipins are localized to lipid droplet surface and can play important regulatory functions, for some of them there is very little consequence if you delete them, surprisingly. And so they do not result in cellular phenotypes, which has led to them being classified as not a hit in this. And that observation for some of the paralipins, such as paralipin 3, appears to be true even in sort of whole body physiology.
Yeah, I would say, just to add to that, there are like 5 paralipins and there's a lot of redundancy. And I think in particular, paralipin 1 is very important in adipocytes, which are not the cell type in the portal. And paralipin 5 is important in cells that oxidize a lot of fuel in mitochondria, such as muscle. So, those things may show up if we get into other cell types and add them to the portal.
Thank you, Maria. Those are the only open questions right now. So I'll just mention to the audience, please post any additional questions in the Q&A, and Maria, please continue with the next section that you mentioned.
Okay, sure. Yeah, we can always come back to the Lipid Droplet portal. Okay, so let me start this. Okay, so as I mentioned, and as I hope at least some of you are aware, the other part of our work as a team is to make knowledge portals for common diseases. And these have genetic association and epigenomic information, and they also have the results of bioinformatic methods that we use to process all these data together and get more insights from them. And so when Bob and Toby approached us about creating the Lipid Droplet portal, oh, I'm sorry, I forgot I had animation here. So these are we have a lot of portals now for different diseases. I'm not going to go through them in detail, but they all have the same basic framework, and they all contain genetic and genomic information. And so when Bob and Toby approached us about creating the Lipid Droplet portal, it was really something different. We did not have these types of data in these portals. And so it really got us thinking about how to handle these types of data. And it also got us thinking that, you know, there is a need for different kinds of portals. And so we started thinking along these lines, and meanwhile, other groups started approaching us, both within our own, you know, group of collaborators with data types that we did not have in the portals, and also just other groups. So I'm going to show a few examples of these. So here's an analysis by our collaborators at the Broad, led by Miriam Udler, who they looked at genetic associations, so variants that are associated with type 2 diabetes, and then they clustered these variants with regard to their associations with other related phenotypes. And so this type of clustering data can be expressed as a heat map, and we didn't have this kind of data or display in the portal at the time, but it was very relevant. We also began to get some complex lists of genes and evidence from collaborators. So a lot of the work in genetic association studies right now is to take the results and integrate them with other types of data, and from that generate a list of the potential effector genes for a disease or a trait. Now, the genes that might actually cause or be directly involved in development of that disease or trait. And so these lists are typically really complicated. This is only part of a huge spreadsheet that has different categories of evidence for these predictions. And then within each category, there are different types of evidence. So we needed to display these as well. And then, yes, there are other data types that people approached us about. This is RNA expression. So we don't currently show that in the portals either. And so that can be expressed as a volcano plot. So we just began to realize that there was really a need for this. And so this is our sort of our universe of what we do. The Knowledge Portal Network is sort of the umbrella that we call everything, and it's powered by this software infrastructure that we call the Human Genetics Amplifier. And so one of our arms here is the Common Metabolic Diseases Knowledge Portal, funded by the Accelerating Medicines Partnership, and there are some disease-specific portals under that umbrella. And then additionally, we have some other knowledge portals that are in the same framework with similar types of data, but they're not directly under the Common Metabolic Diseases umbrella. And we're counting the Lipid Droplet portal in that because it's really a full-scale, fully mature portal at this point. But then to address the needs that I just talked about, DK in our team really developed this whole service that we're calling BYOR, Bring Your Own Research. And this is a framework that people can use to basically upload their own data and configure it how they like and visualize it in many different ways, all without needing any computational expertise. You don't have to be a software engineer. And the displays that have been created using this framework have sort of run the whole spectrum of so portals where we were approached by collaborators. Basically, we built the interface, and that. So, for example, the APOL1 portal would be in that category. And then there are other portals, such as this catalog of VQTLs that were built really just by the collaborators, with maybe some advice and help from DK, but it's people are able to do this on their own. Some of these displays are integrated into our larger portals and some are not. So yeah, just to reiterate what I said, some of the displays are in our KP Labs page. We have a menu of tools that are sort of in beta version. They're not fully integrated into the portals yet, but they are of value, and we want people to use them and give us feedback. So some of them are listed here. We have a whole bunch of predicted effector gene lists that are built on this framework, and those are in the portals. And then, yes, some of the portals are completely standalone. This APOL1 portal started as a supplementary portal with a submitted manuscript. And I guess, yeah, the manuscript is not published yet. It's still, supporting the manuscript, and then this portal could grow as more research gets done. The same for this one. This was some work done by other collaborators at the Broad. So our guiding principles in the service is that you can represent your results just as you intend. And there are core capabilities. We are happy to collaborate with you to construct the portals, but they're easy enough that many people choose to just work on them themselves. And we really value these collaborations because people who are working on portals often come up with new features that would be helpful, and they're valuable to everyone. So we're happy to work on building those. They're easy to use. Yeah, you can stage access. You can set access to be just private to yourself or shared with a distinct group of people or public. So if you need to keep something confidential for a while, you can do that. And yeah, you can leverage the huge AMP resource as you do this. You can link to the larger knowledge portals. So since, yeah, I'm going to take just a couple minutes to show you where you can go for more help on this. Let's see, let's go back to the web. Yeah, so if you go to the Knowledge Portal Network homepage, which is kp4cd.org, there's a link about BOR. So you can click on this link, and here's a page that gives you a lot more information. So there's a white paper giving all the details, and a tutorial and so forth. The tutorial is really helpful. It's very detailed. So you can just work through this and find out everything you need to know about creating one of these portals. And there are, I think, 6 different visualizers at this point that you can use. You can put your data in a Manhattan plot, a volcano plot, and heat map, as I already showed. There's also a region plot, so where you can display genetic associations across a region. So there are lots of options here. So I would encourage everyone who is, might be interested in creating a portal like this to get in touch with us and talk to us about how you can get started. And that's all I have on that.
Thank you so much, Maria. This is really, it seems like an invaluable resource. I don't see any other questions in the Q&A. So and then maybe in the interest of time, unless others have something to jump in with, we will stop there and thank all 3 of you for your contributions today. And it's exciting to think about where this will head. So thank you so much.
Thank you. Bye, everyone.